Hello everyone, Mrs. Hanson here one last time in our Chapter 22 journey. We're going to pick up our topic where we left off, translation and protein synthesis. And we were discussing codons and looking at the genetic code to find 3 bases that specifically code for the individual amino acids. And we had looked at very carefully the transfer RNA. And if you recall, that was looking like that 4-leaf clover where the transfer RNA had this area where we had anticodons down here, and then we had this part right up here that actually carries the specific amino acid. So just quickly, if I scan back to bring that visual back to your mind, and if I here it is, this picture from your notes, kind of cleaned up perhaps. I wanted to bring this back into your mind so that when we start talking about the process of carrying amino acids and building a protein, you could visually see in your mind where this is occurring. Remember this acceptor stem that codes for a specific amino acid? And down here you have the anticodon, which is the translation of the amino acid code. All right, so that's where I'm picking up. I just wanted to have that visual in your mind as we talk about this next slide. So we said there were 3 different types of RNA. One of those types we've looked at specifically called transfer RNA, the little 4-leaf clover. We have a messenger RNA, messenger RNA, that contains the sequence of the codons, and that determines the order of the amino acids in the protein chain that we're building. Now, what brings those amino acids are the transfer RNA. It actually brings the specific amino acid by reading the messenger RNA's codon. And then the site where this is all occurring, where the proteins begin to get built by joining amino acids together, is in the ribosomal RNA. If you recall, that was little r, ribosomal RNA. And it contains all those binding sites that provide the platform for which these proteins get synthesized. So let's really emphasize, when you see a messenger RNA, it contains the codon. And the codon is the very specific 3 letters that give us the amino acid. So for example, UCA is a codon. We went into our genetic code, UCA, and remember how to read this. If the first letter is a U, second one is a C, and then the letter A, that is going to code very specifically for an amino acid called serine, S-E-R. So these are codons. What type of RNA contains the codons? It's the messenger RNA. Please know that. Now, the messenger RNA containing the codon gives me the specific amino acid that we have to go fetch. But remember, it's the transfer RNA that goes to get the amino acid out in the cytoplasm. So it is going to have to read the codon. And it does so in that area that's called the anticodon. So do you remember where that is on that 4-leaf clover? Just bringing it back to your mind, here's the anticodon. It has the complementary code from the messenger RNA. All right, so those are complementary strands so that it can read that genetic code and bring back the correct amino acid. So the anticodon is the complement to the codon. UCA, it's complementary base pairing. A pairs to U, G to C, and A to U. So the complementary code on the transfer RNA would be AGU. But the codon is what I read in our genetic code chart to find the amino acid. Read the codon. The anticodon is the complementary strand, and then we build the amino acid. Okay, so let's really practice that. Each transfer RNA contains the anticodon of the 3 nucleotides that is complementary to the codon in the messenger and identifies the individual amino acid. So messenger RNA has the codon. Those are the 3-letter base sequence that gives me the amino acid that it's coding. So this is from the genetic code chart. I'm reading the codon to get the amino acid. The transfer anticodon is the complementary strand to the messenger RNA, and that is going to actually carry the amino acid, the individual amino acid, back to the ribosomal RNA where the site of synthesis occurs. So let's practice that. Here we have a little practice problem that says, write the anticodon and then tell me what amino acid it's coding for. So think about this. This is the messenger RNA. These are the codons given to us. Alrighty. So on the messenger RNA, we see the codon, CGG. What would the anticodon look like? And to complete that chart, tell me what amino acid it's coding for. Now let me model the first one just so we get an idea. Letter A says the codon that's on your messenger RNA are the letters CGG. That represents 3 nucleotides. 3 nucleotides. The first is the base cytosine, then guanine, CGG. The anticodon are the complementary base pairs, base pairs. C goes to G, G goes to C, so the anticodon would read GCC. We read the codon to get the amino acid. So what is the amino acid that codes CGG? Well, that has to come back up in our genetic code chart, CGG. The first letter is a C, the next is a G, and the last is a G. So I can locate that base right here, that base sequence of CGG from the codon, and it represents the amino acid with the code ARG. So we've identified the amino acid to be arginine, ARG. I'm going to encourage you to pause the video and finish these examples on your own using that genetic code to find what is the amino acid being coded for from the codon and what would the sequence on the transfer RNA anticodon read. Pause, think time, come back. Okay, you've come back, so you must be ready. We mentioned that these are the codons, so we know that those are on the messenger RNA. That these are on the transfer RNA, these are called the anticodons. They're the complementary base pairs to the codon found on the messenger RNA. And the amino acids we represented, I wrote in the left-hand column. How did you do? In letter B, your complementary base strand for anticodon would be CCC. We read the codon for the amino acid. That came out to be glycine. GGG codes for GLY, the amino acid called glycine. Letter C, the anticodon, would be AGG, but UCC is the codon, and we use the codon to code for the amino acids, serine. AUA has a complementary base, UAU, and it was isoleucine, ILE. Letter E coded for proline, and letter F, alanine, ALA. And again, just checking those anticodons, the base there of the transfer RNA carrying the amino acid back to the ribosomal RNA for the site of synthesis. Let's emphasize how this occurs just kind of in a visual way. Remember that we have the anticodon. So here you have the anticodon on the transfer RNA. It's the complementary base pairings for the codon that's on the messenger RNA. The messenger RNA contains the codon. The anticodon is its complementary base pairing. And remember, this section of the transfer RNA is where the amino acid gets carried. So we have an initiation code, we have the segments begin to growing. And as we begin growing the peptide into a polypeptide, into a full-blown protein, they become millions and millions of strands long. So these are very big molecules. Now, the transfer RNA, see how it's bringing the amino acids by the code right to the ribosomal RNA where we have the protein elongate. So the three main parts of translation are shown on this page. We have an initiation code, the very beginning of the protein synthesis. The protein grows through the process of elongation, where one amino acid is brought just by reading the code from the messenger RNA. Amino acids are brought based on complementary base pairing. And finally, we get a code that's going to terminate the protein synthesis. Remember, we said that there were 64 possible codes. 61 of those coded for amino acids and 3 coded for the termination of the protein synthesis. And so when you see a termination code appear in the messenger RNA, the protein is released from that ribosomal RNA, and the protein then is synthesized, and the process is done. So initiation begins when a messenger RNA actually binds to the ribosome, and a transfer RNA brings that first amino acid, always using the codon to read for the amino acid. The codon is our genetic code. And that's what we're reading in that chart with the 3-letter sequence. So an initiation occurs when we have the messenger RNA just bind right onto the ribosome. The transfer RNAs are going to bring the individual amino acids from the cytoplasm to the messenger RNA. Remember, the messenger RNA has the codon, the transfer RNA has the anticodon. You read this to find this. Elongation proceeds as the next transfer RNA molecule delivers the next amino acid. And we learned that's called a peptide bond forming between 2 amino acids. That was back in our protein chapter. And that just continues over and over and over again. The protein will elongate each time a new, just kind of hooking together by those peptide bonds, until finally you read a termination code, and the termination code stops the process. We said that there were 3 codes. UAA is a termination code. UAG is a termination code, and UGA is the 3rd termination code. When you come across those codes, such as any of those 3 there, on the messenger RNA, you know that the protein will be terminated in terms of building it, and then therefore it's released. So when we think about the whole process of coding, remember that the genetic information is stored on a DNA molecule. Remember that we read what's called the template strand. Of the 2 strands in a DNA molecule, one is read, one is not read for the protein synthesis. The template strand always begins from the 3' to the 5' end of the nucleotide chain. When you see a DNA informational strand, it is not used in protein synthesis. We only read one of the 2 strands, and it's called the template strand. The messenger RNA is the complementary base pairing from the DNA to the RNA bases. So remember that if you see a T in the DNA or an A, it gets replaced with there. So remember that uracil is in the RNA, and just kind of remember that you have A, T, C, and G's in the DNA, but you would have A, U, C, and G's in the RNA strand. So the messenger RNA gets built from the template strand of the DNA. The anticodons are the complementary base pairs from the messenger RNA, and we read the codons for the amino acids. So let's highlight what the codons are. I'm going to highlight those in green. Those are the 3-lettered sequences of bases representing the codes for the amino acids. AUG codes for met. UUG codes for leucine, glycine, alanine, glycine, serine. I read the messenger RNA to build the polypeptide. The anticodon is the complementary strand to the messenger RNA so that it knows what amino acid to go fetch and bring back to the ribosome. Alrighty, so let's process that. What polypeptide would be synthesized from the following template strand of DNA? So keyword here, this is the template strand of DNA. This is the coding that's going to build the messenger RNA. We will have a transfer RNA that has the anticodon. The messenger RNA contains the codon, and then we said the amino acid that is going to get formed, in terms of going to code. Oh, Linda, I'm trying to write and talk at the same time and it's falling apart. Amino acid is what I'm trying to write, my goodness. That I'm going to read the codon for the amino acid. Alrighty, so let's try this together. I know that a 5' to a 3' complementary strand going from DNA to the messenger RNA. C codes to G. G goes to C. G goes to C. So the first codon I've just created is GCC, the complementary base pairing from a DNA to an RNA. Now, the anticodon would be CGG, but I read the codon to go get the amino acid. So what does GCC stand for? I have to keep scrolling up. GCC, sorry. GCC, right here it is, and that's the amino acid for alanine, ALA. So we did the first one here, and that's coding for ALA, the amino acid. All right, what does the second codon look like? Well, T pairs with A, G pairs with C, T pairs with A. So here's the next codon that we've just created in the messenger RNA, ACA. The anticodon, A pairs with U, G pairs with C. Oops, I'm sorry, I said that. G pairs with C pairs with G, A goes to U. But I'm going to read ACA for my amino acid. And again, I have to scroll back. You can flip back to that page much quicker than I can scroll, I'm sure. I want ACA. Here's the second letter, C, A, C, A. Right here, ACA, here it is. And that's going to code for THR. Threonine is the name of that amino acid, THR. Here's the next section on the DNA. C would code to G, T goes to A, T goes to A. So here's the next codon on the messenger RNA. The anticodon, complementary base strands, G goes to C, A goes to U, A goes to U. But I'm going to read the codon to find the amino acid. Can you find that one for me? GAA? You're doing it faster than I can, I'm sure. I want to find GAA. GAA. Here it is. That's glucine. GLU. Here it is. And then your final messenger RNA, T goes to A goes to, or T goes to A, and then A goes to U. So here's the final codon. The transfer anticodon would be A to U, A to U, U to A. And what is that last base? We just built one where the messenger RNA contained 3 codons, GCC coded for ALA, ACA coded for THR, GAA coded for GLU, and finally AAU coded for the amino acid ASN. We had 4 nucleotides building that polypeptide. There is a next example in your notes. It says, and now these two. Please pause the video and do the same strategy in the next 2 examples, and then only come back when you're ready to check. Welcome back. Let's see how you did. These are the template strands of the DNA. Notice that the 3' to the 5' is the template strand of the DNA. The messenger RNA is built from the template strand of the DNA, and that's going to contain the codon. And then we read the codon to get the individual amino acid sequence. Did you build ARG connected to VAL, connected to ALA? LEU, repeated twice, ending with serine, SER. The complementary base strands are used to build, remember I should add that this 5' to the 3' end on the complementary base strand for the messenger RNA. And now check also your ARG to GLY, to PHE, ILE, VAL, and ASN. The skill here that we're looking at is noticing that we read what's called the template strand of the DNA. From there, you'll be able to build a messenger RNA that contains the codon. You may be asked for the anticodon on the transfer RNA. And then the individual amino acids use the genetic code, those 3 bases in a sequence of 3s. That genetic code gives you the individual amino acids. Remember that you read a codon to code for the amino acid, and you've got this section down. In the next sections, you have some assigned reading to do. You're going to look at some mutations. What happens if you have a misspelling in a genetic code and the possible genetic diseases that may lead to it? So you're going to look at things called mutation, mutagens, and look at the different kinds of mutation. You'll see as you go through your reading, you could have a point mutation, a deletion mutation, an insertion mutation. And as you go through and you realize the effects of each type of these misspelled genetic codes. And the last here you'll read about is called a silent mutation. No effect at all there. That happens more frequently than we could ever be aware of. Then you'll look at some different combinations of these genetic mutations and the diseases that they cause, such as sickle cell anemia and so forth. And then the next section you'll read about is recombinant DNA, and look at the 3 key elements to form a recombinant DNA. And you'll go through some of these structures together here. So the last part of it is really just assigned reading. I look forward to receiving your note packs. Let me just tell you, it's been a personal joy serving as your instructor this term. This is going to be our last video together in our journey of organic chemistry. Have a good day, everyone.